Hello, everybody. I'm very excited to have you all here for a live look at some amazing science and restoration happening out on a big ship in the Gulf of Mexico right now. Our team members are about 150 miles off the coast of Texas and Louisiana, making their way to a site that is over 1,000 meters deep or 3,280 feet. My name is Sasha Francis, and I will be your host today. I'm the Education and Outreach Manager for the Mesophotic and Deepwater Communities projects, working to restore deep seafloor coral habitats in the Gulf that were injured by the 2010 Deepwater Horizon oil spill. Those are some big words and quite a mouthful, so we're going to kick things off by talking about what that means, what habitats look like down in the deeper parts of the Gulf, and why some of them have needed our help. So after a quick intro video, we will get connected to the scientists at sea shortly. We're so excited to have you all here to see what it's like to be on a ship for days, or in this case, weeks at a time, and conduct important science and restoration work. Today, we'll be taking a deep dive into how animals are able to survive and thrive in the deep sea, and how we're learning more about these animals on this expedition. So please share any questions or comments that come up in the chat. We'll pause for questions as we go. If you're not able to type into the chat, no worries, you may have to log into a YouTube account to be able to do that. Please do that now. All right, so as you go deeper into the ocean, like the underwater robot you see here, a remotely operated vehicle or ROV, less and less sunlight is able to penetrate the water. Around 160 feet down, you reach the mesophotic zone, which literally means middle light, and it's sometimes referred to as the twilight zone. As you travel even deeper, you'll eventually reach a point where there's no sunlight at all. In addition to this change in light, the water also gets colder and there's much higher pressure. Despite the low light, cold temperatures, and high pressure environment, there are incredible ecosystems that call the seafloor at these depths home. Colorful corals, sponges, they're the foundation of these ecosystems, providing habitat for the many fish and invertebrates you see here. These deep sea ecosystems are found all over the world, including the Gulf of Mexico, which is where all of this beautiful video footage is from. There's still so much we have to learn about the deep sea because exploring these depths can be difficult, and it requires expensive, highly technical equipment and unique skills. However, we do know the deep sea is the largest habitat on Earth. The number of species and abundance of life that depend on these habitats may rival the colorful coral reefs found in shallow waters or tropical rainforests on land. Unfortunately, the Deepwater Horizon oil spill in April 2010 dealt a heavy blow to Gulf communities on land and at sea. 11 people lost their lives, and natural resources, wildlife, and habitats across the northern Gulf were severely impacted. Restoration is particularly challenging for the deep coral habitats that were injured because of how far from shore and how deep they are, and because of how much we still have to learn about them. As a result, 4 restoration projects are currently underway to help us better understand and restore these seafloor ecosystems from 160 feet to thousands of feet below the surface. These 4 projects are mapping new areas and developing better maps of the seafloor in the northern Gulf of Mexico so we know where these coral habitats are found. They're learning more about species that live in these habitats and how they may be connected across the Gulf of Mexico. ROVs like this one are particularly helpful to get incredible visuals of what lives at these depths that humans can't always reach. We're also collecting samples and testing different methods to help corals grow and reproduce in the wild and in labs in order to help replace corals impacted by the oil spill. We're going to be learning a lot more about that today. And we're working to reduce threats that these ecosystems face and connecting this restoration work to people like you. This work is made possible by the Deepwater Horizon Open Ocean Trustees, including National Oceanic and Atmospheric Administration, or NOAA, the U.S. Geological Survey, or USGS, the Bureau of Ocean Energy Management, plus many more partners, including a large number of academic institutions and non-governmental organizations. We'll be talking to some of those folks today as well. So we're about to connect with the ship. And as we do that, I want to note why this mission is particularly cool. This is one of the first significant NOAA-Navy collaborations, it's a long word, in the Gulf of Mexico. It's the deepest dive by humans for a NOAA science and restoration mission, and one of just a handful of times that saturation diving has been used at sea for a NOAA mission. So we'll talk a little bit about that and a lot about the coral and specimen collections that are going on right now. All right, let's see if we can see folks on board. Ooh, little quick pick at the awesome ship they're on. And I will be going out tomorrow to join them at sea, actually, on that massive ship. And I believe it's about 394 feet long, which is definitely bigger than any vessel I have been on. Oh, here we go. Hey guys, how you doing today?
Good.
Awesome. I see that it is Fun Shirt Friday. Is that right?
Yeah, on science missions, we often celebrate Aloha Friday, wear Hawaiian shirts.
I love that. We do have some really cool habitats out there in Hawaii as well. All right, so the work of restoring habitats on the seafloor of Gulf of Mexico takes a lot of people with lots of different jobs. So, can you introduce yourselves and tell us where you're from and what your role is in this expedition?
All right, sure, I'll go first. I'm Mark Laytonen. I am a collections technician at the Smithsonian Institution, National Museum of Natural History in DC. We're one of the many partners in this MDVC program, and I am here to, first and foremost, preserve any of the specimens we bring up and bring them back to the museum where they'll be archived as a permanent record of the biodiversity we're encountering. And we also have a lot of scientists at the museum who are interested in doing sequencing work on stuff we bring back and putting better taxonomic IDs on it.
Very cool.
Hello, everyone. I'm Jack Gerard. I'm coming from the University of Rhode Island, PhD student there in Carlos Prada's lab. And in terms of NDVC, a couple chapters of my dissertation focus on that coral propagation techniques thing. So essentially a little bit of coral farming or coral ranching, whatever analogy you want to use. So I'm putting down settlement tiles and fragmentation racks. I think we'll get into that a little later. And I'll give it over to Matthias.
Hi, everyone. My name is Matthias Gomez. I recently finished my PhD in Dr. Carlos Prada's lab at the University of Rhode Island. And my role here has been supporting Jack with his project. Mostly what I study is coral population genetics. So I've been supporting some of the identification of the corals, soft corals mostly, through genetic means, and also supporting propagation methods, just kind of building structures so that we can propagate corals in the deep sea.
Fantastic. And I'll just note that the University of Rhode Island, where a couple of you are from, is my alma mater. I went there a long time ago, but I'm very excited to be working with y'all, and URI is helping support this awesome opportunity to connect with you at sea. So very excited to call back to them. All right. So many of us are probably most familiar with shallow habitats, shallow corals, things you see maybe when you're at the beach or snorkeling or even recreational scuba diving. Can you tell us what's different about these mesophotic and deeper habitats? Like, what do you see down there that's different?
You go first.
Sounds good. Yeah, so it's the big thing with the mesophotic is, as the name implies, meso middle, photic light. So there's a big decrease in light. And for shallow water corals in particular that are in association with an algae that actually allows them to use that light to make food and sugars and that sort of thing, the challenge then becomes how do you derive energy using less and less light? So some species that can live in the shallow, we'll say shallow seas, that move into the mesophotic, you start finding that their skeletons change or their entire shape changes. So if you're familiar with brain corals, normally in the shallows, they're mounded, they're rounded, they do very much look like a brain. But as you get deeper and deeper, you'll find that they start plating out and looking more like a dinner plate, or they start having more of a skirt instead of a mound or a hill type of shape. So that's one way.
You'll also encounter a lot of more soft corals in the deep than in the shallow, and so shallow corals tend to be flatter and more boulder shaped because they're just trying to catch light. But as you go deeper, the primary source of energy for corals is catching stuff out of the water, so catching marine snow or particulates from the water. So you'll see fans like you're seeing now. These big corals stick up in the water and just kind of spread to cover as much surface area as possible to catch the current as it passes by and try to sift particulates out of the water.
Yeah, and I think we also have a video of what it looks like when they eat, which Jack was mentioning, and you were just mentioning, Mark. The way they eat is quite different. We can see it a lot more easily, the way that they're grabbing the food out of the water column. And I think one of our coral labs actually has a little video clip of that to see it up close, which is very cool. But that's really important to note is how different it looks. Oh, there it is. And I will say, when we've done that, we showed this clip at the Smithsonian broadcast we did, and the people in the audience were mimicking the coral polyp eating like this. It was very cool how it kind of reminds me of the wacky waving inflatable arm flailing tube man. It's very, very fun to watch, and it's really, really cool to see them actually, grabbing the particulate from the water and eating it. So what do they eat? When y'all are seeing them and eating in the water column, or we're feeding them in the lab, what is it that they're eating?
Yeah, so I guess in many ways all sorts of things. Some of it is dissolved nutrients that are just in the water, be that carbon or nitrogen. But the stuff that you're seeing them grab, I guess it could be phytoplankton, it could be like zooplankton, copepods, that sort of thing. Some of that I think is dictated by the size of the tentacles at the end of the polyp and that kind of thing. But it's I don't know. What we got?
May change as they grow too.
Yeah. Anytime you see footage from the deep sea, there will just be a constant rain of like this white particles that looks like dust falling down. We call that marine snow, and that's basically just any tiny organisms like dying near the surface just slowly sink down, and things that like when things go to the bathroom and stuff, it just all sifts down to the bottom, and basically things that live in the deep are grabbing that stuff and sifting through it and getting the nutrients where they can.
The great circle of life. Love it. So are there any other adaptations you want to mention for different creatures that are able to live in the deep sea that you can think of?
In terms of bigger animals like fish, often what you'll see is sharks are a good example of this, is counter shading, where a big animal will be dark on top so that if you're looking down, it'll be harder to see because you'll be seeing darkness below you. And if you're looking up, if you can still see light, it'll be lighter. So things are dark on top and light on bottom, and that kind of helps camouflage them from predators or prey on either side.
Yeah, that's really cool to point out, because I think people might be familiar with that, seeing dolphins, for example, at the surface here in the Gulf of Mexico. They'll see that difference as well.
Another thing, especially when it comes to color. So right at the surface of the water, you have, we'll say, all the colors that we can see, and the farther down you go, you don't lose the colors evenly. You lose them pretty much from red to blue, especially out here in the Gulf or in other like coral reef environments. So when you start getting deeper and you see corals or really anything becoming more and more red, the reason that they're red is because we're adding the light. When you don't add the light and you were to just basically see them as they are, they're a lot more drab and they're a lot harder to see. So in that sense, a red coloring can be indicative of, instead of this bright, vibrant thing, actually a more camouflage type of lifestyle.
Wow, I never knew that. That's really, really cool. Very neat. So when we think about corals, we like I said, we mostly are connecting those with the shallow water coral reefs. And I know that we see that kind of as like a reef structure. But as you go deeper, it kind of looks more like a forest, right? It's more like a community of different corals. So it does also appear differently, right?
Yeah. So a lot of, like I said earlier, a lot of the corals, instead of being like flatter and like mounded to catch light, are sticking up into the water, and kind of each coral in a way becomes its own reef. Like all the other smaller animals tend to make their homes on those corals sticking up into the current as an easier way to get into the current to catch those particulates. So on a lot of big corals, you'll see things like basket stars or squat lobsters or all sorts of little critters that are living on that coral to use it as a way to get into that flow.
Yeah. And then on the other end of that, as you create that structure, or you have those organisms that want to grow up, right, you've now created a whole bunch of spaces that other things can live in. Stuff to hide from predators, build homes, and that then ends up being, in a larger context, what makes things like corals or soft corals or any of that so important, because it becomes so much of a habitat for everything else that it then kind of drives the system, right? That it's, if you think of a forest on the land, right, without the trees, a lot of the stuff that lives there wouldn't be there in the first place. So you need that as the foundation, if you will.
Yeah, that's a great point. We mentioned that in the introduction, talking about how these habitats are that foundation. And one of my favorite things I've learned working on these projects is calling the the other creatures that like to live on the coral, associates, like they work for a law firm. That's been one of my favorite little anecdotes. So when we're studying the deep sea, tell us about how you might use technology like ROVs to learn about and help restore these habitats. I know we have a really big one on board the ship. So how does that work in contributing to this?
Well, I mean, just to start, an ROV helps us get down there to depths that we can't easily go ourselves and to collect samples. The ROV, the technology is pretty impressive these days with the dexterity of the arms. So we can often collect small pieces of the corals without destroying the whole colony, and we can often pick up even other animals that can move around and try to get away, like crustaceans and stars. But it's very cool to be able to bring this stuff up from the traditional methods. Like historically, how we've gotten these samples has been from trawls, where they just drag a net across the bottom and you would get stuff, but you would also destroy a lot of habitat, and the samples that would come up would be quite badly damaged often. So we get to see live color video of these animals still alive, and we get to selectively and less invasively pick up specific and targeted animals. So that's a big part of it. But we can also use it to help our other efforts, like the coral propagation studies.
Yeah, so it's a huge privilege just to be able to get down there and see it and take some of the samples. But as soon as you have to put anything down there or try to do any kind of manipulative or mensurative experiment, that kind of adds the difficulty. So, I mean, just a shout out to all the guys here on the Island Intervention. These pilots are super skilled. They have an incredible machine. Sometimes it can take a really long time to cut samples or place things or move stuff at all. But just, yeah, there's a lot of talent here, and I've been very appreciative of it.
Yeah, something else to note about when you're using the ROV is you can also get some water samples up with that, right? Because when you place a specimen or an item into the bin on the ROV, it will come up with some water from that same depth, right? Is that helpful for y'all to get any other information? And if not you, probably some of the other scientists, but.
Yeah, actually, we know that these organisms rely on what is suspended in the water. So by collecting water samples at different depths, we can actually study what the nutrients are. Also, we can look at the temperature and many other organisms that we don't normally see just by the naked eye. So that's also really helpful for us. And something else that I wanted to add is that actually making natural observations of these organisms on the water is really cool, because many of the times we are just looking at organisms that have been preserved. So looking at what they really look like down on the water is something that is really cool for us. And it's also very informative for scientists.
Yeah, that's amazing. I know that our coral lab scientists, aquarists that we have in our coral labs, have used, water samples throughout these different expeditions also to really try to nail the parameters and things and learn more. Since like I mentioned when we started, there's still so much we have to learn, which is an important part of this restoration work is filling knowledge gaps. So going from ROVs, I know some of you mentioned the dexterity can be challenging with those, even though the pilots are extremely talented. So we also have some specialty divers on this expedition. And I was chatting with Jack earlier about how neat it is to see them at work compared to the contributions the ROV can make. And it's really cool to have both opportunities on this expedition for the coral propagation and outplanting racks. So Jack, could you talk a little bit about that and how they're helping with the coral propagation work?
Yeah, absolutely. It has been amazing to have both things. So part of the experiment or the restoration work really is assessing which methods sort of work best. And now one can imagine that if you have to cut a coral, bring it from the deep up to the boat, get it into the fragmentation racks, and then send it back down, that's pretty traumatic. So although the corals are generally pretty hardy, and so far we've seen some good success, another way to do that would be if you could do the whole thing down in the bottom, something we would consider like in situ or within the environment fragmentation. And because the divers, right, they have their hands and they have such amount of bottom time, at least in this type of saturation diving, it really gives us the opportunity to kind of do this in a much less traumatic way for the coral. So as an example, Matthias and I, the night before the divers did this fragmentation, we did it ourselves, aided by the ROV. I think we ended up turning 3 corals into about like 56 fragments.
Wow.
But the divers were able to take 56 different corals and turn them into 56 different fragments without having to bring any of them up. And they did the whole thing in about 2 hours. Matthias and I, it took us about 24 straight hours to get those things up, cut, and back down. So it was really amazing yesterday to have that just go so well, so smoothly. And I mean, just looking at it, like it's a pretty fascinating operation. So yeah, huge win yesterday.
That's very cool to have the aid of the saturation divers be able to try things, by hand and at depth. And I know that some of our first coral outplantings, they were able to bring them up, in the ROV collection bins, keep them underwater, keep them in that temperature and in those parameters, and then make fragmentations still underwater and place them out in the fragment racks. And those are still doing well based on the technical diving team. We've been able to go out and check on them, but it is really amazing to be able to try these methods at depths and see what kind of different things we see down the line with the growth rates and whatnot down there. So really cool stuff. So I wanted to pivot a little bit to we're also looking at marine debris on this trip, and I'm sure y'all have seen some neat stuff come up out of the water. Let's share for a moment, why is marine debris an issue for habitats in general living in the deep sea?
Part of it, or I'll cover one aspect. First thing, if you have marine debris down, very rarely do things stay still under the water. Yes. Especially given storms. So there's always a risk if something is down that it gets picked up. And if you have corals or any sort of structure, that thing just becomes a wrecking ball. So that's one thing.
Yeah, and often the debris that is lost is stuff like fishing gear or large like scientific equipment attached by line, and that rope and line can get wrapped around reefs and corals, and if something snags it and pulls on it, it can do quite a lot of damage in a very short amount of time. And also various debris with electronics in it or batteries can leach like harmful chemicals into the environment for those animals. But it's also like animals on the bottom like structure, so debris also in a way provides habitat, so there's always going to be stuff growing on it. And so we're also interested in pulling it up to see what's made a home of it. And also, if when the debris was lost, when recovering it, it's kind of an interesting time point. Anything that's living on it or growing on it can only have been there for however long. So it's an interesting part of this is like it's picking up trash, it's getting a closer picture at a big habitat. Like a lot of, as you're seeing some pictures of us picking through debris right now, and you can see that big buoy is entirely like encrusted with a rainbow of colors, and that's going to be a lot of encrusting animals like sponges and coralline algae and tunicates. And those are things that are pretty hard to collect with an ROV because it's basically flush with the surface and like really on there. So this gives us a chance to get a closer look at some of the smaller stuff and a lot of the like weirder, flatter, encrusting animals that live down there.
That's amazing. And, I hadn't really thought about the movement of large marine debris on the seafloor. I think folks don't really necessarily think about that. We're used to seeing here on the surface, maybe plastic water bottles or something like that. But yeah, even really big, heavy things on the seafloor can move a lot with the currents under there. So that's a really important note. So we have just another minute or two before we'll pivot to questions from the audience. But I wanted to ask y'all, what is the coolest deep sea animal you've seen so far on this expedition, either on the ROV camera or, coming up for a specimen collection?
Well, you go first. Or if you need time to think. I mean, I'm a crustacean enthusiast, so we've brought up a couple sponge crabs, which are weird, small little grumpy looking crabs. They have a very like angry face, and they wear a sponge on top of them. Yeah, that's one right there. We took his sponge off, but yeah, he had a big sponge, kind of like I can envision it like a hermit crab, except instead of a shell, he's carrying a big piece of a sponge as protection and camouflage. And we brought up a couple of those, the same kind of crab but with different sponges. And I just think they're adorable and a pretty cool symbiosis there.
Ooh, looks like an arrow crab. These guys are pretty
crab.
goofy looking.
We've been bringing up some good crustaceans.
Yeah, they've all looked angry.
Yes, most of them do kind of have a natural angry face.
I guess for me, I think one of my favorites that you've brought up was actually that massive basket star that we got the other day. Every now and then you see a couple of them, on the shallows, but I'd never seen one so big and just kind of looking this way. It just, I don't know. You look at how spread out it is, and you just wonder how it can manage all those limbs.
I guess I'm gonna be very boring here, but because most of the time I'm seeing preserved organisms, so for me actually to see live corals. So Swiftia excerta, one of the species we've been trying to restore, just to see that thing like moving and feeding and just the colors it has been mind-blowing for me.
Yeah, that's a very good point. Like a big part of why I'm here as well is to take color photographs of the specimens when they come up before preservation, because like most taxonomic research involves scientists looking at preserved specimens. But when you put something in ethanol, the vast majority of invertebrates lose all their color. So they become white, they bleach out. And so like, here's a good example. This coral was bright yellow. After being put in ethanol, it is now black. Like crinoids, this is a crinoid, a sea lily. It was probably bright red or yellow. My colleague collected this while I was asleep, but they're often bright colors, but then they just kind of go a muted brown or white. Crustaceans all turn white, and you saw earlier some of those photographs of beautifully red and clear shrimp, but then after a few days in ethanol, they'll just be white. So it can be taxonomically informative to see the colors of an animal, and you only really see those when they're coming up alive or when you're looking at video on the bottom.
Yeah, that's amazing too, to be able to have the ROV footage to go along with your specimen collection. Like you said, if you're, collecting blindly, it can be, really challenging to get the whole picture, I think, which is an important part of this restoration work is, filling those knowledge gaps, like I mentioned, learning more about these deep sea habitats, that there's still so much to learn about. And every new thing that we learn, every, specimen that we can collect and work we can do in our coral labs to learn about their reproductive rates, et cetera, observing them and on propagation racks out on the seafloor. All of this is really instrumental to contributing to the greater restoration of the habitats on the seafloor in the deep Gulf of Mexico. So thank you all for all your work contributing to this restoration. I want to pivot over to take some questions from folks here on YouTube. So we'll see if we have any questions coming in here. All right, let's see. Someone said they love the coordinated outfits. Not a question, just a comment. So someone asked, and we can, I think, Mark, you can take this one. Would the specimens being collected predominantly be going into the invertebrate zoology collections at the National Museum of Natural History?
Yes. So some corals are going home with, a couple went to the Audubon Aquarium in New Orleans to be put on exhibit there. A couple are being taken by some of our coral propagation and husbandry labs to try to keep alive and research like how best to keep them alive. But by and large, most of what we're collecting is coming home with me to the Smithsonian, where the vast majority of what the Smithsonian has is kept behind the scenes. You're seeing a clip now of like the storage space in the Museum Support Center where most of our alcohol collections are housed, but we have millions of specimens for researchers to come visit and look at, and we also can send specimens on loan. So it'll all be cataloged and added to our collections and made available. The records will be public eventually for people to look at, and they'll also be able to see the photos by searching our collections portal online. But then the specimens will also be available for research, and they will be a permanent part of the collection and useful to scientists down the line. And we have active postdoc researchers looking forward to some of these specimens. We've been sending pictures home. We probably have new species. We tend to find new species on almost every expedition. So there's a lot of interesting stuff coming up.
Incredible. That's really cool that not only are they going to be, useful for informing this restoration work, but also for scientists to work with and learn from down the line, and different folks throughout that network of scientists. It's incredible. So are any of the specimens y'all collecting also going to Prada's lab and aquariums and things like that throughout this expedition?
Yeah, we sent a big Swiftia colony and a couple Mauricia corals, soft corals, with an aquarist who was on one of the earlier legs of this cruise. Those will be at the Audubon Aquarium in New Orleans, and then the Prada lab. Are you guys taking any?
Not on this one, but in the past we have. So for some of that genomics work that we do, or other things of, I also use the word phylogeny, but essentially figuring out how different things are related. We will take some soft corals for that and send them back. And I think Matthias has actually been heading that project a little bit. But otherwise, on this cruise, we're just trying to get them back down there and grow them.
We have another colleague taking some corals back to the Etnoyer lab in Charleston to try to keep them alive, some other species.
Amazing. And yeah, for those listening, the Swiftia is that really bright orange one, and the Mauricia that they mentioned is the big white octocorals that you saw some footage of on the seafloor. So are coral propagation techniques for deep sea corals significantly different from shallow water coral propagation? I don't know if any of you have experience with shallow water corals as well, if you want to speak to that a little bit. I know a lot of what we're doing, it's kind of the first time it's ever been done with these deeper corals.
Yeah. So I personally haven't tried to restore shallow water corals, but having lived in that world and read some stuff, a lot of what we're doing is informed by that. So it's kind of variations on a technique. Although I will say a lot of the shallow water coral stuff I think is a bit farther ahead than where we are. This is very much kind of a proof of concept. So fragmentation is something that's done in shallow water corals, outplanting, that kind of thing. So we're just trying to modify that. How do you do that down deep where you can't basically send a typical diver? So again, where you start needing ROVs, where you need, so the clip you're looking at right now. So there's we'll say active restoration, which is the fragmentation where you're taking corals and you're cutting them up into different pieces from the original and trying to plant them out and around the reef. The other thing you can try to do is create and leave artificial habitat, hard substrate, so like a hard bottom stuff. So this is a whole bunch of settlement tiles that we've made out of concrete. And the idea there is that we can set those down and try to track the amount of settlement or recruitment that happens. So the way corals propagate is they'll put out larvae, and that's going to swim around in the water column, sometimes for days, sometimes for months, depends on the coral and the conditions, and eventually they will settle onto a space. So by putting these tiles down, we have a way of measuring that and assessing that if we were to leave the space alone for a certain amount of time, how long do we think it would take for it to naturally restore itself? And in there, from a restoration perspective, you can start making decisions based on, do we need to intervene and take a more active process, or can we kind of let it alone and let it restore on its own as long as we protect the space and prevent further degradation?
Very cool. And there was a question about how deep are they? How deep are we going today, for example, or have we been kind of going this week with the different coral work and marine debris removal?
Yeah, so for the mesophotic stuff, we really haven't done too much below 100 meters at this point, so about 333 feet, if I have my conversions correct. So that's been kind of where we've been about for the divers and for our restoration stuff. But I think where we're on transit to now is much deeper. I think you mentioned at the top of the show, we're heading down to like 3,000 feet or like 1,000 meters or something like that. And I think a lot of the other researchers are going to be putting down different kinds of landers, and they're trying to be pulling up sediment cores. Again, a lot of that habitat assessment. What are the environments like? They certainly could speak to it a little better than I could, but really just trying to collect all that information so we can understand what organisms are facing, and again, getting at those like challenges of these spaces.
Yeah, so I think we've been mostly kind of more in that mesophotic zone now. which about in the 300-foot range or so. I know everything past 160 feet is what we're looking at. And then, like I mentioned earlier, that we're traveling right now. They're transiting now to those deeper sites. So when I get on the ship on Sunday, I know we're going to be focusing more on those deeper depths with ROV video footage, etc. So very excited to see that in action. And I know the saturation divers will be decompressing at that time. Won't get to see them dive, unfortunately, but very cool to see it all in action. Fantastic. I think we might have one more question before we wrap up here. Oh, this was a good one here. Follow-up question for specimens at Smithsonian. Will there be any 3D scans?
Probably. That's a relatively recently gotten off the ground, but we've had a couple people working on doing 3D scans of certain collections, and I'm not in charge of what like the priority list, but I think we definitely want to 3D scan as much of our stuff as possible and make it available. We all think that's a really cool thing that we're doing, and we're happy to keep doing more of it.
Yeah, I was trying to look on my desk here. I have a small 3D printed Paramaricia from one of the deeper corals, and the 3D photos being taken at the Smithsonian. So I know they're doing some of the 3D photos to be able to provide some more, complete images of that. That'll be very, very cool. All right. Well, I think that's all we have time for today. And well, we do have a question about live streaming any ROV dives. Yes, if you are on the YouTube channel right now, if you click back on that live button when you're done, you'll be able to see which upcoming ones we have left. So we have another broadcast tomorrow and then one on Monday. We won't really be able to live stream just the ROV footage, but we will have clips from those ROV dives as they're happening during our broadcasts. So stay tuned for that. I think that's all we have time for today, but we please feel free to check out our website so that you can dive a little bit deeper into this restoration work. And I want to thank all of you for joining us here on the ship and spending some time with us today. I know you probably have wacky sleep schedules and have been working very long days. So I very much appreciate you taking some time with us today and supporting the restoration work out in the Gulf of Mexico. We really appreciate all your contributions, and I'll see some of you soon. So thanks for you joining us. Thanks for folks here on the YouTube joining us today. You can learn more about deep sea restoration in the Gulf of Mexico by visiting our homepage, which they're going to drop into the chat. I think it's already up in the chat already, but keep a lookout for that. And then if you do have a moment, please give us some feedback. There is a link in the chat as well for our feedback form for this experience. Please let us know what you thought. And we'll see you soon. If you enjoyed today's event, spread the word to friends and family. Check out those upcoming events on the University of Rhode Island's Inner Space Center YouTube page that I mentioned. If you just click back to that live channel, we have a few more programs left on this expedition. And we'll have more programs coming live for missions happening later this year in September and October. So stay tuned.
Right.
Thanks
for having us.
joining us. Ooh, second camera angle. Beautiful.
Oh.
Happy Fun Shirt Friday.